Hello everyone, and welcome to the online Biological Sciences Seminar Series of TÜBİTAK Research Institute for Fundamental Sciences, which we organized to mitigate the negative impacts of COVID-19 coronavirus outbreak on scientific thoughts and science-based actions. It is my pleasure to let you know that I have the honor of introducing our today's speaker, Professor Zhang, a very well-known world-class scientist from the Department of Computational Medicine and Bioinformatics and Department of Biological Chemistry at the University of Michigan. He is going to give a great talk on protein structure prediction. As usual, at the end of the talk, we will have a session for questions, which can be asked by using the chat button of the Zoom platform. The research interest of the Zhang lab is in protein design, protein folding, and protein structure prediction. The iterative threading assembly refinement, or ITASR algorithm developed in this lab, was ranked as a #1 method for automated protein structure prediction in the worldwide CASP competitions in 2006 and 2020. Professor Zhang is the recipient of the Delano Award of the American Society for Biochemistry and Molecular Biology, and the Alfred Sloan Award, the US National Science Foundation Career Award, and the University of Michigan Basic Sciences Research Award. Professor Zhang was selected as the Thomson Reuters highly cited researcher. With this, I want to thank Professor Zhang once again for joining us for this seminar and look forward to his talk. Zhang, please, you're welcome to begin your talk.
Thank you, Alikram, for the nice introduction. And yeah, I also want to thank Dr. Turkan Haliloglu for the very kind invitation, which gave me the opportunity to share with you guys about the recent research we have done on the protein structure prediction. So this is really the right time to talk about this problem, because recently we have seen very exciting or revolutionary progress of the protein structure prediction, which has been brought about by the new technique from the deep learning. So this is the reason I put the title is deep learning has really significantly impact protein structure prediction. So before I go to the talk, I like to, or I know not everybody here are expert on this problem. So I will try to spend a couple of minutes to introduce background, like what is protein structure, why we are working on this. So this is a slide showing the very well-known so-called central dogma of life. So we know that every of us will have a genome which include, by the way, can you see the mouse of my, the monitor of the mouse?
Yes, it's okay.
Very good. So every of us, we have a genome, and then there's heredity information is deposited in the DNA sequence. And through the transcription and translation, those DNA sequences can be translated to protein amino acid sequence. Then the amino acid sequence will fold into a unique shape of protein. So through that unique fold, and almost all the biological or cellular function is carried out by the protein molecule. So here is one example, is called myoglobin, which is help us to carry the oxygen from our lung to the muscle. So here you can see this picture. There's the red part is the oxygen molecule. The bond carried by the heme and the histidine group, and the heme group itself is carried by the very well formed excuse me, the binding pocket. So to carry this function, that unique shape formed by the myoglobin is a key role here. This actually is a general rule for the protein molecule, and their three-dimensional structure determine their biological function. So solving the protein structure will be very important for us to understand the function of protein, and also guide us to to design new drugs, because any of the small change of the protein sequence or molecule or structure, they can cause very many human disease. So when we talk about the protein structure determination, there's a long history, and there's many people's effort. So probably the first milestone can be traced back to more than 100 years ago, when Dr. Zhang discovered the X-ray. And then later, Dr. von Loy, he found when the X-ray cross the crystal, they can form the diffraction pattern. And then in the 1958, Dr. Zhang, he using the X-ray diffraction to solve the first protein structure. Is just I mentioned, is a myoglobin. And then later there is a NMR, it's a relatively new technique involved. And about 10 years ago, there is a cryo-EM is also become a very interesting and important technique for solving the big protein molecule. Almost all this protein structure which we solved is deposited in a database. It's called the Protein Data Bank. That is formed in 1971. So if we I just checked yesterday, and the PDB, now we have nearly 150,000 protein structures. So if you look at the trends, you can see this x-axis is year, y-axis is the number of the protein structures in the PDB library. They are increasing very fast, and nearly every day we have 40 new protein structures that have been solved. Very encouraging. But if you compare the number of the sequences, which is usually in the UniProt, compare with the structures in the PDB, you will be frustrated, because here, look at the scale is different. Here is 1,000, and then y-axis here is a meaning. So compare with the sequence, the structure looks like not a change, it's really ignored. So if you compare with the number of the sequence and the structure 13 years ago is about 100 times the gap. In 2010, it's 200 times. 2015 is 500 times. Until now, number of the sequences is more than 1,000 times more than the protein structure. But we know that the sequence itself cannot tell us too much about the knowledge about the protein function. So that really tells us that the number of the structure can never catch up the sequences. Basically, it's because decide a protein structure is much more expensive experimentally compared with with designing a protein sequence. So what we can do? So this is a question we were asking. Given a sequence, whether we can write a computational program to predict or calculate this three-dimensional structure. So this is one of the major challenges in the modern computational biology. So that's also the topic we're going to discuss today. So let's first look at what we have done in the last about 50 years about protein structure prediction. And the protein structure prediction generally can split into two categories. One is called template-based modeling, and one is called ab initio folding. So template-based modeling means you can identify the template or homologous structure from the PDB library. And then you can use that homologous protein as a template to guide you to build the full-length model. And you have two ways to identify the template. You can either use comparative modeling. Basically, this is a query sequence. And then you know the PDB structure. They have also their sequence. You just compare their sequence alignment. You can see which protein have the highest sequence identity. And then you can use that template as a guidance to build your model, because here the assumption is that the homologous sequence, if they have a higher sequence identity, they have higher the structure similarity. But there are so many cases we don't have a good template, have close homologous template. The sequence identity can be low. Then people use a technique called threading to identify those non-homologous template. So here, instead to search the sequence space, they directly compare the query sequence with the PDB structure, and then scoring them and identify the best non-homologous template. So the ab initio folding is different because they don't use a template. So in principle, you have to use some like physical rules, like to calculate the atomic interaction, and then use some emphasis assumption that to find the lowers the free energy state, which we suppose is a native structure. So this is a summary of these two methods. Usually, if you have a good template, through the comparative modeling, you can build a very close the structure to the native. So sometimes you can reach to below 2 or 1 angstrom, and that is very close to experimental resolution. But if your sequence identity is lower, then the threading can still build a correct fold, but the accuracy is slightly lower. Generally, if you don't have a good template, and then you build the model from ab initio folding, your accuracy will be lower and it's not very reliable. So those are traditional way for modeling, and the categorized into different category and method. Also, the different model you have different functionality, which you can help use this computational model to annotate. So just Alikram just mentioned that we developed a method is called ITaser, which is we try to combine the 3 different methods. So we're starting from a sequence, then first we do threading or comparative modeling to identify the template, which we hope we can help us to build the model. But in many cases, the template can be unreliable or can be incorrect. So here, this is one example. The thick backbone are the native, the aligned region. and the same backbone are online region. So in iTaser, we try to remove those online region and then reassemble the continuous fragment. Using Monte Carlo simulation, we build the full-length model. You know, when we run the Monte Carlo simulation, we can generate many different decoys. You know, the conformation. Not all of them are close to native. So to select the native structure or near native structure, we do cluster. Here, one point is one structure conformation. And then the biggest cluster corresponding to the lowest free energy state. So then we select the conformation from the biggest cluster. The next step, we try to rebuild the model, you know, split this, we call the cluster centroid into the fragment and reassemble them. And then we select the lowest energy state to get the final model. So that is a general pipeline for iTaser. But like every other program, you know, if you build a protein structure folding algorithm, you need a 3 component. First, how you represent your model or protein conformation in the computer. So in iTaser, we represent the user reduced model. This is a 4 atom model. And then we use reduced conformation, means each residue represented only by the C alpha and the cytosine centroid. And then we project them on the lattice so that we can search the conformations faster. The second component is energy first field. So we don't use, you know, our first field is mainly knowledge based on the, you know, statistics from the PDB structure. And we also optimize the weight factor based on the correlation between the decoy arms D and their energy. So the final component is how you search your conformation and how you select the model. We use a replica exchange Monte Carlo simulation, and which can help us to pretty quickly to search the conformation. So to test their the performance, we collect a pretty big or the largest benchmark data set at the time when we developed the method. So a very important part is that we try to compare our model with the template. So this is a very important problem because here I show you like during the CASP competition, and the assessor has been constantly pay attention to how the final model compare with the template. But the conclusion is that very disappointing that similar to what has been observed in previous edition of the experiments, no model result to be closer to the target, means the native structure than the template. So generally at that time, when you don't move the template or you make it worse. So this is the right hand side is what iTaser is doing. So here x-axis is the RMSD of the best template. Y-axis is the final model. So this probably is the first large-scale benchmark test which is showing that we are able to draw the template closer to the native structure. And one of the major reasons we can do that is that we are able to combine multiple templates to build the final model, because some of the template which you cannot have the good quantity for this template in this region, but can be catch up by other template. So that's a major advantage of iTaser. Like a modeler, this is very often used software in the community. They are mainly based on single template. So you can see majority of the cases is just the point stated in the diagonal line, which showing that the template is not really refined. So this benchmark shows that we are, be able to fold about 2/3 of the protein if we make a cutoff, like if we suppose 6.5 angstrom corresponding to a correct fold. So it's higher than the model. But still, we are asking why we are not able to fold the rest of the 1/3 of protein. So to figure out that reason, and here we are, this is an ITSR pipeline. We start from the template by threading. So we try to ignore the threading template. We use our, native structure to search the PDB library to find the best template. And then we want to check what we can do. So the result is quite surprising. We show that if we are able to use the best possible template, we are able to solve the protein structure prediction problem, because nearly 100% of the cases, we can fold the protein which have an RMSD below 6 angstrom. So that gives us a hint that, if we use real threading template, we are like this. But if you use the best possible template, we can fold like the curve like this. So that highlights the one way to solve the protein structure prediction problem using the template-based approach. So the major challenge is how we can identify those templates which is close to native, but are non-homologous to the target. So that is what we talk about the ITSR, because they are based on template. But we also develop a method called Quark, and that can help us to fold the protein without using homologous template. So the basic idea is that we can split the target sequence into the fragment and then match the secondary structure from the non-homologous PDB structure, basically random structure. Then we use that fragment as sample guided by a physics-based force field. And the major new idea here in Quark is that we can use the fragment to identify those long-range contact. And that can help us to guide our fold the global structure. So we are not the only lab which develop different protein structure prediction problem. So here there is just a short list of some well-known laboratory, those PI's name, the institution software name, and the methods they are using. There's many others, the method we are not listed here. So the question is that, which method is working and which method not working well. So to answer that question, and we are, have this community, we organize a so-called Olympic game in protein structure prediction. It's also called a CASP, C-A-S-P, which like Alikram just mentioned. So in every 2 years, the organizer, they collect about 100 protein sequences, which nobody knows the proteins, their native structure. And then they send these sequences to the people, the predictor. You run your program and see what is the protein structure you can model. And then in the same time, they will send these sequences to the experimental structure like X-ray crystallographer, MR, or nowadays is cryo-EM, asking to solve the structure. So 3 months later, there's an independent team, scientists, they are comparing the predicted model with the experimental structure, and then to check, can objectively to check which method is working, which method is not working. So this is a very important experiment, which is the first is in 1994, and every 2 years you can see this number of groups. From 2000, there was a solver group is introduced. Solver group means you are the structure are modeled with automatically without the human intervention. So we, ITASER, we participated in the CASP in 2006. So then, until CASP 14 is in this summer. So there's some milestone, starting from CASP 12, there's a new technique is called direct coupling analysis based on contact, which I will talk about later. So in 2018 to 2020, there's a deep learning technique introduced. So I will mainly focus on this recent forecasts and highlight what are those very exciting progress has been made in this, or witnessed in the CASP competition. So in CASP 11, those are the number, how many human expert, number of the automatic solver and the target domains. So here, based on the structure or the availability of the template, the target can be categorized in the two groups. One is the template-based modeling, and one is the free modeling. So for template-based modeling, one of the major goals is to try to build a model which is closer to the native compared to the template. So this is what we have seen. And here, again, the x-axis is the armsd to the native of the best template. And the y-axis is the armsd of the first model. If you are one point here is one protein. If you below the diagonal line, means you are, refine the model, refine the template. The right-hand side is a TM score. TM score is higher, is better. So you can see majority of the point are above the diagonal line, showing that we are really able to refine the final template, closer to the native. So that is the template-based modeling. Probably the more interesting cases is the free modeling target. For those one, you don't have a template. So you have to use some physical base or some other method to build the model. So this is probably, two of the most successful free modeling examples, which we have found before the new technique is called DCA and deep learning has been introduced to the field. So in these two cases, we don't really use any homologous template, but to build the model, you can see pretty close to the native. Left-hand side is X-ray structure. Right-hand side is a predicted model. This is another target. So we can say this is one of the target which is folded by quark, and this is the closest structure template which exists in the PDB library. You can see they are completely close to the target structure. So why we are able to fold that and to show that this is a, if we use iTaser to assemble the threading fragment, you can see iTaser, they are doing better than the template, but they're still not the correct fold because the TM score is 0.4. But when we use the Quark, basically we reassemble the N-terminal tail, and then using the medium range contact predicted by the segment-based contact, we are able to build the model which is very close to the native structure. So those in CASP 11, we are able to fold 3 target, 3 point, have a TM score higher than 0.5. Here 0.5 is a cutoff, and we found that if your TM score higher than 0.5, your global fold is close to the native structure. But you can see, we still have some of the target, the correct fold have a length is marginally higher than 100 amino acid. That is cutoff, for most of the ab initio folding method. So this is showing some progress we have made compared with previous CASP. So what about CASP 12? In CASP 12, you can see this is the x-axis is the length again, y-axis is the TM score. You can see we have folded a lot of more of the model which have the TM score higher than 0.5. So why we are doing so much better? If you look the success rate, use a cutoff TM score higher than 0.5, there is a jump from 10% to 34%. The reason is that we have a new technique to develop the contact. That's called co-evolution. The idea is actually pretty simple. If we give a protein sequence, you can identify a number of the homologous sequences. And if one point, for example, here is I, here is J, use these 2 positions, there are 6 amino acid, correlated and mutated, then it's likely they are interact with each other or they are in contact. So here you can see if R is not changed, then D is not changed. If R mutated to a K, then D mutated to a E. If K mutated to a W, then E mutated to a V. So this correlated means in the space, probably those residues, there's some interaction. Otherwise, they will not like that obvious correlation. So that idea was proposed by the Dr. Sander and his colleagues, but they are not, that proposed more than 25 years ago, but they're not working pretty, particularly well. One of the reasons is that there's a transition noise, because here is one example. If A and B are correlated mutated, and B and C also correlated mutated, then A and C will be correlated mutated. But you can see A and C themselves is not in direct contact. So if you just based on the mutual information, there's some noise in the contact. So later, especially like 10 years ago, the Dr. Wick, they proposed a new method which we can remove the noise from the correlated mutation. So I will not have time to go into detail, but the general idea is using the maximum entropy model to express the probability using a partition function, then this EIJ will be directly related with the contact. So that significantly increase the contact accuracy. So what we did is that we are using that method and integrate into the naive-based classifier and also neural network to further increase the contact accuracy. And then we put the contact map into Quark, which we have pipelines called C-Quark, contact-based Quark. That help us to significantly increase the contact, final model. So those are we check 8 successful protein folding in the folding target cases in CASP-12. We can see 5 of them are due to the contact map prediction. So Quark itself also folds 3 cases. The contact is not correct, but they can still fold the protein. So what about CASP-13? In CASP-13, the major change is that we introduce a convolutionary neural network. So that is a very typical new technology is called deep learning. So starting from a query sequence, we can learn and how the protein are amino acid in contact. Then we can put the contact into Quark and iteration. So here the critical part is how we train the deep learning into the contact map. And we first collect the multiple sequence alignment, then calculate the both metrics, convergence metrics and the precision metrics, then put it into the deep neural network. And we found the accuracy is much higher than what we previously obtained. So this is like 10 years ago, we used general, very simple deep learning, machine learning method. So the accuracy is about 20% long range. So those are 2 methods using the co-evolutionary contact map. So this is a newer method for the also using deep learning. So you can see the new deep learning method is much higher than previous method. So here show that how deep learning impact the structure prediction. Again, y-axis is the deep learning based Quark modeling, TM score. X-axis is the old Quark method. We don't use contact. You can see the majority of the cases we have the TM score is higher than the model without a deep learning contact. This is iteration. So especially this region, they are very encouraging because we don't use a template, but we can still fold the model which have the structure accuracy close to the template-based model. So this is the CASP-13. Again, the x-axis is the length, y-axis is the TM score. And we have a lot more of the target. Now we have 32 free modern target in the CASP, but we can fold 23 of them, which have an accurate, overall success rate is 72%. So if you look at the history, you can see from CASP-11 to CASP-12 and then to CASP-13, there's a jump of the modeling accuracy. So the reason is that, again, I just emphasize in CASP-12, we have a co-evolution. CASP-13, we have deep learning. So CASP-14 is this year. So in this year, we have using the query sequence, and then we also use deep learning, but we train on both contact and distances, also train on the hydrogen bonding network plus torsion angles. And then we put into ITaser. This is called D-ITaser. So this is a new program which we are releasing. So one of the very important things is that we predict the distances, and the distance is split into bins. Here the axis is the distance of the ratio I and J. Y-axis is the probability. Usually they have follow a Gaussian-like distribution, also have a cutoff. We usually set a 20 angstrom. Then we convert the probability into energy using this equation. This equation we can nicely convert the different situation of the distances into the energy function. So we also combine the multiple predictor from different regions to guide the ITaser simulation. So here we show the final, the newest model by the D-ITaser compared with the original ITaser. And this is the TM score of the D-ITaser compared with the previous in CASP 13 ITaser. So we still have a pretty significant improvement compared with like 2 years ago. The major reason is that we have introduced distances and which can have more detailed information which we can learn from the PDB library. And this is compared with the original ITaser. You can see this part is really very encouraging. And there are many of the template, non-template based target, which we can fold really have an accuracy close to the experimental resolution, especially for those ones which we have a TM score higher than 0.9. So this is a comparison of the CASP 14 compared with 13. So you can see we can fold here all the target again. They are free modeling, means they don't have any template which exists in the PDB library, and we can fold some very big single domain protein with pretty high accuracy. So those are the what we can say the 10 best solvers, automatic solvers in the CASP 14 on the 97 domains. And this is a GDT score. This is a Z score. GDT score is is a matrix which is used by the CASP assessors to assess the model. And you can see, let's say here the blue trace is a model and the red trace is a native structure. The GDT score compare the distances of every atom pairs and then look at the number of the residue have a distance cutoff, for example, D below 1 angstrom or below 2 angstrom or below 4, below 8. Add all the three number of the residue pairs, the different cutoff together, divided by 4L. So if you have a perfect model, your GDT score will be 1. So those are the GDT's overall GDT score. And this score shows that how significant of your model compared with the average group. So the Zhang solver is basically is ITaser, and the quark is what we talk about, the D quark. So this is what we have seen in the, we participated in the CASP in CASP 7, which takes place in 2006. And this is every 2 years we participate. You can see the ITaser worked pretty well compared with the other state-of-the-art method. But we can see the gap previously, originally the ITaser and other methods, there's a pretty big gap. But since CASP 12, especially since new technology was introduced, and the gap between our lab and others become smaller and smaller. So yes, if I don't mention this, really the AlphaFold 2 is not complete. So the most exciting breakthrough has been introduced or witnessed by the, in the CASP 14, which is achieved by the AlphaFold 2, is developed by the Google's DeepMind institution. So they are building a model which, have majority of the cases have very high resolution. And they are, about half of the cases, they have a GDT score higher than 0.9. This is a typical example to show that the blue is the AlphaFold model and the gray is the X-ray structure. You can see they are very close. Amazingly, they don't use any template. They just use deep learning to learn the trained in the PDB library. This is amazing because this topology is so complicated. They can model, nearly perfect of all the detail of the native structure. So this is the AlphaFold 2, their architecture of their machine learning neural network. So this is a summary which I copied from the Dr. Hasbis's talk. They highlight what's a key difference between the first version AlphaFold in the CASP 13 and the AlphaFold 2 in the CASP 14. So the major difference is now from the previous, they train the distances, like what we are doing, and then use the gradient descent method to build the model. Now they use machine learning, use end-to-end training, direct from the sequence to the three-dimensional structure. So the second new technology part is they are using the attention-based neural network instead of the convolution neural network. There's some other, the new point. So that is is a remarkable breakthrough. People think now probably for at least for the single domain level of protein structure prediction, and that problem can be solved by the neural network or deep learning. So now we come to the conclusion. And the first conclusion I'm happy to draw is that we can now have a paradigm shift from structural template-based modeling to the sequence-based machine learning. So previously we are built the model mainly based on the template or based on structure in the PDB. Now we need to collect the multiple sequence alignment and then can use machine learning to model the very high accuracy structure. So the shift is from relying on the PDB library to relying on the genome sequence. This is really a very big change since, which the field has been trying very hard in the last 50 years. But there's still a challenge. So first, how we can derive the multiple sequence alignment from the sequence database, because now there's many very big genome databases, and they are increasing very fast. So there's a meta-genome or whole genome database. Not the bigger, the better. So the point is that you need to collect those highly relevant MSA so that you can trace back, accurately about those co-evolution information, buried in the sequences. Second, we need to develop very sensitive deep learning model to develop a model from the, model sequence alignment. They have a low number of effective sequences. So for our lab, we are working on two things. One, we combine the deep learning with ITaser, which is one of the best or more advanced method to assemble the three-dimensional structure. We are also trying to use the cryo-EM low-resolution data to combine that with the ITaser, because that also, we found the preliminary results show that using low-resolution cryo-EM data, we can also build a very accurate model. So finally, I want to, thank our colleagues or members in our lab, which is do all the work which I have talking about. So we are mainly focused on 5 categories. So today we talk about the protein folding and the structure prediction. We also work on how we can derive the, do the functional annotation based on the structure, or we derive the protein-protein interaction network in the structure. So protein design is a category which we are, very excited and put a lot of effort on that, because we try to use the method or force field we developed for protein folding to design new sequences. Also, we spend effort to study the mutation, because many of the mutation which is caused very serious, human disease. So we try to predict what are the mutation which cause cancer and human disease, but what are other sequences which doesn't. So I also want to thank this, this foundation agent which give us support. So thank you for your attention.
Yeah, thank you so much for this very nice, deep and exciting talk. Can we take now some questions through chat button?
Yes.
Please, questions.
Let me check, how can I?
Or you can ask just by raising hand. So we have one questions. Can you see it, Yang?
I cannot see that. You can probably.
Chat, please check chat button.
Okay. During your presentation, I realized that most successful models are all obtained from relatively small protein. So I wonder if we can obtain successful prediction model for relatively bigger protein structure as well, or is this a very advanced stage of the structure protein by deep learning? Okay, yeah, this is a good question, which is reflect also related with the progress of the field. If you can look at this, in ancient time, actually not that ancient, like 10 years ago, we can not fold big protein. So you can see the successful, when we don't have a template, we can only fold the protein previously below 100 amino acid. So in CASP 11, we can marginally fold the small to medium size. But you can see now, when we use deep learning, we can fold a very big protein which without a template. So this is pretty remarkable, because if you come to a protein like 400 amino acid, it's a single domain. Their topology is very complicated. but we can still fold them. That shows that the power of the deep learning. When you learn the contact, the distance map, especially, this is a contact map, this is distance map. Also dihedral angle, hydrogen bonding. The structure is already there. So ITaser here is the task is try to assemble the structure based on that information. Of course, slightly refine the region which we don't have a deep learning information. So to answer your question directly, is that now the bigger protein structure folding is not a problem in the using deep learning. And yeah, that is the main attribute to the information, the power of the deep learning method. So I saw the second question, do you train the machine only with the parameters which are in the PDB files, or do you provide additional information for the machine to evaluate externally and help the assessment process? Yes, first of all, we are trained this deep learning based on the PDB structure. So we collect a whole representative set of the single domain PDB structure. I think they contain about 26,000 of the PDB structures. And then we train based on the multiple sequence alignment. And we didn't really specify what are the structural feature, is trained. And where our training is mainly based on the sequence. The feature we are using is mainly from the feature we derived here. There is a covariance matrix and a precision matrix. They are derived from the multiple sequence alignment. And then let this, the deep neural network to learn all these feature or parameter. So we have some parameter for the neural network itself, but there's no, parameter which we have tuned for the structure feature part. So I hope that answers your question.
Türkan Hocam, do you want to make comment or ask questions?
Yes, please. Dear Yang, thank you so much for this very interesting talk. I have a general question. I mean, why we cannot predict the 3D conformation structures from first principles? What are the main challenges or obstacles in that?
Yes, that is the dream. But unfortunately, we are unable to do that. Part of the reason is that if we are talking about so this is talk about the physical based first field, this first field is approximation. They are not probably, work very well for very complicated protein structures, because for some very small protein, and there is some error, but if your majority of your term is right, you can still do something to fold the protein. But we know that their first field is not that accurate. When you come to a very big protein, and any of the small error in the energy term, they can accumulate to a big error. So that's part of the reason. And we cannot use the first principle method to fold the protein, because if your final model or lowers the free energy state is not corresponding to the native, then even you are able to search through the entire energy and then sleep, probably you will still come to the wrong, local minimum. And another part of the reason, those are the atomic interaction for the protein. But the very important part is the solvation, solvation atom and the protein atom in that interaction. That play a very important role in protein folding, but we don't have a very accurate, formula or equation to describe this kind of protein and solvation interaction. So I think that those are the reasons which we are still not yet able to fold the protein based on first principle.
Okay, so another question. Have you tried deep learning in protein structure complex predictions?
Can you say it again?
Have you tried deep learning in the prediction of complex structures? Like protein complexes.
Complex. Okay.
Yeah.
Yes, this is what which we are doing, but we are still the paper not yet published. I think this probably this is what we are doing, and where certainly is a future direction what we are doing, but probably also the future direction of the entire field. Also, as far as I know, that Google's DeepMind, they are also probably this focus of their the goal, how to introduce the deep learning. New technique is so powerful to more biological relevant questions like protein-protein interaction or protein-ligand interaction, or drug discovery or protein design. So to answer your question directly, we are working on that, and we found that deep learning can also very powerful to model the protein-protein interaction complex. So here, the very important part is that how you can derive the multiple sequence alignment from the protein-protein interaction. And the challenge is that we have many sequences for the single chain protein sequences library, but we don't have that many reliable protein-protein interaction sequences. So that will affect the power for the deep learning based complex structure model.
Okay, thank you. And the final question, you use protein structures from PDB in deep learning. I wonder whether using some model structures, wrong structures, let's say, would help in deep learning, like just teach in a way, what the wrong structures do. Maybe it is not a relevant question, I don't know, but I'm just curious.
Yeah, this actually is a very interesting idea, because first of all, all the training we are based on the PDB structure, means this structure is correct.
Right.
And yeah, this is an interesting idea. How we can probably also introduce a decoy, using the so-called negative learning.
Yes.
So that can be useful for us to improve, the recognition power for the correct or incorrect structure folding. But there's some related, the progress on this is that, in the DeepMind or also other groups, they are trying to introduce the error prediction into the deep learning. So that is one of the very important steps. You need to predict or learn what region your model is unreliable. And then those regions, how you can refine that part, make your learning, individual or iteratively or gradually converge to a region which you find, confident in every region of your sequence. That is one of the strategies is which we are working on that, and also other people showing that. But that is related with your question about the, but we never try to introduce a decoy into the learning. But probably we can try that.
Okay, thank you very much again.
Burak Bey, please ask your question.
Okay, thanks, Dr. Zhang. My question is a bit technical, actually. Well, let me, actually, there are two questions, but they are very much related. First, have you checked with the interpretability of the convolutional neural network model? As far as I can see, these are for predictive purposes, and they are great. But what does the neural network, convolutional neural network model does so that it can increase the folding prediction that much better? Does it come up with some new physical features, right? Interpretable features. And my second question is, it is very interesting that as an input, you put both covariance and also inverse of covariance together. So does it really improve? I mean, do you really need to feed both of them at once? If you just feed one of them, isn't this sufficient for the neural network model? Because the other one is just a transformation. Thank you.
Yes, you raise a very important question for the, so the field which is pay attention to. So deep learning, like many other, machine learning method, it is a black box. So give the sequence, train, and then they get the final model or final result. So that's part of the, the shortcoming for the solution based on the deep learning, because we don't really know what's going on inside the box. And we don't know, like, if you do the first principle method, just like Turkan mentioned, then we are able to interpret physics or the folding dynamics of the protein folding. But deep learning cannot tell us anything about that. So of course, we can also try to train the machine learning, see whether we are able to, to train the dynamics. But we currently, we don't have that many experimental data, because the PDB library, they only tell us the final equilibrium structure. So there's no intermediate state for us to train that. So to answer your question directly, this is, we are not able to interpret the physics based on deep learning. But that is, there's some interesting, interesting attempts on this. During the CASP 14, there's some attempts, people try to learn the protein folding, not based on PDB library. Just like, in AlphaGo, there's a Go game. The first version is to try to train the AlphaGo deep learning based on the human Go game, this schemes. So that is based on human data. The second version of AlphaGo, they are trying to train or by fighting by themselves using the rule of Alpha, the Go game. They're doing much better than the first version of AlphaGo. So here, probably there's some attempt. Actually, the DeepMind also tried that to train their model, not based on PDB, but they are not doing it too well. So they are much worse than the train based on PDB. There's some interesting attempt on this.
We have one more question through chat.
Okay, let me.
Can you see it?
Yes, this is from Ahmed.
Ahmed.
Thank you for the presentation. It's great. Do you consider a single correct folding for a protein? Is it a possible alternative form in vivo? Yes, this is also a very relevant question. And in the deep learning, we are mainly training the final model based on the a single stable structure. So currently, we didn't focus on this, the alternative form of the protein structure, because we know that in real cell, and the protein can have alternative conformation. But this is an interesting direction. Probably, if we introduce those different states in the training set, that is possible to address this kind of question. So that's very much depend on how we train our model. Probably, when you do the model training, like when we derive the contact map or distance map, we did see, there's no unique answer. There's a distribution during the histogram. So we do have multiple confirmation if we build the final model. But how that is related with the alternative form, which we have seen in vivo, we haven't done that. But this is an interesting question to check.
Mm-hmm.
So I saw the another question from the chat from Dr. Akbas. Thank you for your great talk. Considering the current achievement, can we expect more groups to incorporate deep learning into their method in the next CASP? Absolutely. I think even in this CASP 14, I didn't see all, but at least all the top 10 groups, they have used deep learning. So that certainly is a trend, and at least in the field of protein structure prediction. But I can imagine, I think this is very obviously that many of the related field will be probably dominated by the technique of deep learning, including, like Turkan just mentioned, the protein complex interaction modeling or drug discovery, especially the protein design. Those one we can immediately see the deep learning will play a very important role.
Yes, it seems that we have no more questions. So Zhang, thank you once again very much for coming and join us for this seminar. We will look very forward to meet you here in person as soon as this coronavirus settles away from the world.
Yeah, it will be my pleasure. And I also, very happy to give this talk, and thank you for all the very insightful questions. Hope to meet you guys next time.
Yeah. Thank you. So we can stop here, yeah?
Okay. Thank you, guys.
Thank you.
Bye. Bye. Bye.